Myself, Dr. R. Sanap, Head and Associate Professor, Department of Botany, Rao Bahadur Naran Rao Borake College, Rampur. Today, I am going to deliver the lecture on the study of Pinus. In that, the subtopic is the TS of or transverse section of the Pinus Needle. It is the part of syllabus of T.Y.B.Sc Botany, paper 4th, that is BO 334, and title of the paper is the Spermatophyta and Paleobotany. In that subtopic it is the gymnosperm, in which here there is a topic that is the study of life cycle of Pinus, and today we are going to see the TS of the Pinus Needle. So here if we see the Pinus leaves, so this Pinus leaves, if we see these leaves of the Pinus, these are needle-like. So if we see this Pinus needle, they develops on the dwarf shoot, and this Pinus, they show the presence of the needle-like leaves. So these leaves, they are not dorsiventrally flattened, but they are acicular. So such type of whorl of the leaves, they are developed, and these leaves, these are the acicular, the needle-shaped leaves are present. And as these are needle-shaped, they are not dorsiventrally flattened, that is having the dorsal and ventral side. So they are acicular or needle-like, and that's why they are commonly called as the Pinus needle. And these are the foliage leaves, and they are doing the function of photosynthesis. They are long-lived, so they remain for much period of time and doing the function of the photosynthesis. So thus here these are the leaves, commonly called as the needle, Pinus needle. So if we take the transverse section of this Pinus needle, then it shows the presence of the different types of the tissue layers. So it shows the presence of, here the outermost layer, it is known as the epidermis. So see, this is the epidermis. So this epidermis, it is single layered. Epidermis, it is single layered. See, this is the epidermis, and this epidermis, it shows the presence of a thick layer of cuticle. So this cutin substance, it get deposited on the upper surface of this epidermis. So this is the cuticle. Cuticle, it is a waxy layer present on the upper surface of the epidermis, and this epidermis, it get interrupted by the presence of the stomata. So this epidermis, it shows the presence of the stomata, and the stomata, they are sunken. So here sunken type of stomata are present. That is, sunken, it is nothing but they are present deep in the deeper region, having the stomatal cavity or stomatal chamber, and that opening, it is called as the stoma. So thus here, this is the small opening, and that opening, it is known as the stoma. Stoma, it is nothing but the opening of the stomata. And inner side to this epidermis, there is a presence of the hypodermis. So here, if we see this hypodermis, so this hypodermis, it is 3 to 4 layer. So the cells of the hypodermis, these are compactly arranged. Cells are polygonal in shape, compactly arranged, and sclerenchymatous. So as the cells are compactly arranged and sclerenchymatous, So their walls, they are thick and they are lignified. That is, the lignin substance it get deposited on the walls of these cells. So thus these cells, these are the thick walled, these are sclerenchymatous, and these are compactly arranged without intercellular spaces or intercellular gaps. So this is the hypodermis, which is made up of the sclerenchyma cells. This is the hypodermis. 3 to 4 layered hypodermis is present, and here there is the presence of the stomatal chamber. So here this is the stomatal cavity or stomatal chamber, where there is a presence of here the stomata. Now here inner side to this sclerenchymatous hypodermis, there is a presence of the mesophyll tissue. Now here this mesophyll tissue, it is not differentiated into the palisade and the spongy tissue. So it is not differentiated into the palisade and the spongy tissue. So the cells of this mesophyll, these are the living, they consist of the chloroplast, and that's why this tissue, it is green colored. And as it is the green colored, so this tissue, it is doing the function of the photosynthesis. So this photosynthesis process, it is carried out by this tissue that is known as the mesophyll. So this is the mesophyll. So as I told you, it shows the presence of the chloroplast. So here chloroplast, it is green colored. That's why that tissue, they becomes green colored, and so this tissue, it is doing the function of the photosynthesis. So thus this is the chloroplast. This it is not differentiated into the palisade and the spongy tissue. And inner side to this mesophyll, there is a presence of the vascular tissue that is having the stellar region. So it shows the presence of the, so thus here this is the stellar region having the different types of the tissue layers. So it shows the presence of the single layered endodermis. So here this is the endodermis. So this endodermis, it is single layer. Cells are compactly arranged, and they are somewhat barrel shaped, and thus here it forms the outermost layer of the still region. So it is known as the endodermis. So this is the endodermis. Now inner side to this endodermis, there is a presence of the pericycle. So here, so this pericycle, so this pericycle, it is actually the parenchymatous tissue. So the cells, they are thin walled. It is many layered or multi-layered, and its function is that it transports the water and minerals from xylem towards the mesophyll tissue. So thus here, this is the pericycle, which is parenchymatous, and it is the multi-layered. So this is the pericycle. And in the center of this stele or the vascular tissue, there is a presence of the vascular bundles. And if you see these vascular bundles, so these vascular bundles, they shows the presence of the phloem which is facing towards the outer side. So here the two vascular bundles are present at the center. So it shows the presence of, there is a presence of the cambium. So this cambium, it produces the tissue towards the outer side. Tissue which is produced towards the outer side, it is known as the phloem. So this is the phloem. So this is the phloem. So cells are phloem, they are living. So this is the phloem. And this is the cambium. So this is the cambium. So this cambium, it is the meristematic tissue. As it is the meristematic, it cut off the cells or produces the cells towards the outer and inner side. The tissue which is produced towards the outer side is called as the phloem, and it produces the tissue towards the inner side. It is known as the xylem. So this is the xylem tissue. So this xylem, it shows the presence of the metaxylem, which is facing towards the outer side, and the metaxylem, it is facing towards the inner side. So this is the protoxylem and this is the metaxylem. So the cells of the metaxylem, they are large in size. So this is the metaxylem. Cells are large sized, thick walled, lignified. This is the protoxylem. So thus here the xylem which is facing towards the inner side of the cambium, and in the middle portion there are the medullary rays. So these are the medullary rays, and in between the vascular bundles, so there is a presence of the transfusion tissue. So this transfusion tissue is present at the central region. So here there is a presence of the transfusion tissue. So this is the transfusion tissue, and in the mesophyll region there are some region canals are present. So in this region, region canals are also present in the mesophyll region. So this is the region canal or the region duct. So thus here, if we see the internal structure of this TS of needle or transverse section of needle, then it shows this such type of the tissue layers having the outermost layer that is called as the epidermis, which is covered by the thick layer of cuticle. Then it get interrupted by the presence of the stomata. So here there is a presence of the opening, and here it is the stomatal chamber. So it is followed by here the hypodermis. Cells of hypodermis, they are dead, sclerenchymatous, compactly arranged without intercellular spaces or intercellular gaps. And inner side to this, there is presence of the mesophyll tissue. This mesophyll, it is all the cells are of single type. It is not differentiated into the palisade and spongy tissue. Here they are green in color as the chloroplasts are present, and that's why this layer it is doing the function of photosynthesis. Here in the mesophyll tissue, raging canals or raging ducts are also present. Inner side to this, there is the presence of the vascular tissue having the single layered endodermis, many or multi-layered pericycle. So this pericycle, it is parenchymatous tissue, and it transports the water and mineral to the xylem elements. So here, this is the pericycle, and here there is a presence of the vascular bundles. So this vascular bundles, so two vascular bundles are located at the central portion. And one more thing is that here the vascular bundles, so these vascular bundles, these are conjoint, they are collateral and open type. Conjoint, collateral and open. So as the vascular bundle, it consists of xylem and phloem. So it is the conjoint. Xylem and phloem, they are facing side by side on the same radius. That is, xylem is facing towards this inner side, phloem is facing toward the outer side. So they are collateral, and in between the xylem and phloem there is a presence of a strip of cambium, so it is the open type. So thus here vascular bundles, they are conjoint, collateral and open type. And here this metaxylem, it is facing towards the outer side, and this protoxylem, it is facing towards the inner side. In the vascular bundle, these medullary rays are present, and in between the vascular bundle there is a presence of the transfusion tissue. So thus here this is about the interstructure of the Pinus needle. So this is about this today's part, that is the TS of Pinus needle. Now here I am going to conclude. Thank you very much.